Hello, everyone. Next, I will introduce our work. It's titled Large Language Model and Ontology-based Analysis of Influenza Vaccine Adverse Events. I'm Wang Zhigang from Peking Union Medical College, and this work was proposed by He Yongqun and Zheng Jie from University of Michigan Medical School. First, let's understand some background information about large language model. GPT-3, 4, and 4.0 were developed by OpenAI. They are most popular models with about 175 billion parameters. Gemini was developed by Google. It's able to handle multiple types of data, including text, image, audio, and video. Both of the GPT and Gemini can only be accessed via web or API, so they can't be installed locally, and the privacy issue should be considered in biomedical research. Llama 3 is a powerful large language model developed by Meta company. Is capable of text generation, classification, and question answering, and is available in two versions, one with 8 billion parameters and the other with 70 billion parameters. Gamma 2, developed by Google, is a character, is highly effective and with transparent parameters, so it is well suited for fine-tuning. LLM in binding is the vector or arrays that can represent semantic meanings of word, phrases. After we convert the text to vector, we can use cosine coefficient or Pearson correlation to calculate their similarity. And the text in binding is able to emulate the linguistic intuition. So we can see these sentences are similar in the linguistic intuitions. And it is important to identify the vaccine adverse events pattern and the underlying mechanisms. And the vaccines are often associated with various AEs. We can get the AEs from FDA package insert file because this file usually records such AE information. And the wires also provided the structured information of AEs reported by doctors and patients. This study, we only use the FDA package insert. Ontology is a formal representation of knowledge, including concepts and their relationships. And here are some ontologies can represent the AE's concept. For example, HPO describes and categories phenotypes associated with human disease. Measure is the default AE classification of virus, and OAE is ontology of adverse events. After we get ontology information, we can use the semantic similarity to compare them. For example, two genes can map two lists of geoterms. The association of the genes can be calculated via the geo-based semantic similarity. And the overall hypotheses of this work are: the first, LM can understand the FDA package insert and find the VAEs from the text. And ontologies and LLMs can be combined to support enhanced vaccine AE study. So we want to evaluate how LLM can support AE extraction and AE association analysis, and then compare LLM and ontology-based semantic similarity analysis method. Our workflow has 4 parts. The first, we use the Llama 3 large language model to extract AEs from the package insert of the FDA-approved vaccines. Second, we use LM text embedding to convert AE text to vector. And third, we will develop an automatic tool to map vaccine AE text to ontology. In this work, we use HPO ontology. And fourth, we compare LM-based text embedding and ontology-based semantic similarity method to analyze association between vaccines. And this figure is the workflow of our work. Here's some detail. This page shows the URL where we get the data of the FDA-approved vaccine package insert. And we choose Llama 3 large language model to extract adverse events section. The Llama 3 with 8 billion parameters can run smoothly on my local machine with minimal computing resource. My computer has memory of 22 gigabits and a graphic card of NVIDIA RTX A2000 and 22 gigabits memory. Also, the Gamma 2 with 27 billion parameters can run on the machine like this. And this page shows the original PDF file of the package insert file. And here is the adverse reaction section of this file. And we just use our prompt to extract this section. Here is the prompt. Use the data package insert content, extract the text under the section adverse reaction without adding any introductory phrases or sentences, and response based solely on the provided data. Do not give response using your own knowledge. The red text is the key of the prompt. We use MX by embedded large model to convert the text to a vector. The AE text convert to 1024 dimensional vector. And we use Pearson correlation coefficient to compare pairwise similarity of vaccines. And based on the embedding vector, we do vaccine clustering. Scene binding can help in machine translation, text classification, and recommendation system, and the RAG-based knowledge-driven text generation. RAG is retrieval augmented generation, while the text embedding model is primarily used in retrieval documents or text fragments related to the question you ask in RAG. In this work, we utilize it differently. We just use this model to find the relations between AE text. We also developed an automatic tool to map AE text to ontologies. And this step can help standardize VAE concepts. And this tool was based on the LLM and the RAG theory. After this step, each vaccine was associated with a set of multiple HPO terms. And then Lin's method, which was based on the information content, was employed to calculate the similarity of the ontology sets. And this result shows the clustering result based on these two methods. Left is the result using the LM embedding method, and the right is based on the ontology similarity method. We can see the left finger shows more differential vaccine profiles than the right finger. And most of the vaccine similarity in the right finger are very similar with high correlation coefficient. The majority of clustering patterns are similar, and by cut tree, the vaccine generally falls into three categories. And also we can see this vaccine is flu mixed, both in the left and the right finger, as stand out as being significantly different from the other vaccines. And this vaccine is a live attenuated vaccine. DSNE is a visualization technology that can reduce high dimensions to 2 or 3 dimensions. After we reduce the 1024 dimensions to 2 dimensions, we can see the scatter plot of the vaccines. The distribution of the vaccines are aligned with the K-means result. A different color represents the classification assigned by K-means clustering. Also, this vaccine flu mixed is stand out. After the paper submission in this meeting, we use a similar method to study all the vaccines available at the FDA. There are 111 vaccines. We manually annotated the features for each vaccine, including the vaccine's indication, whether the vaccine is recombinant or not, whether it's live or not, is it a mRNA vaccine or not, or monovalent or multivalent vaccine, or adjuvant use or not. We can see the clustering result are aligning with the vaccine's indication. Here, the green vaccines are the influenza vaccines. Also, we can see many of these vaccines are clustered together. Also, we can see in this column, many live vaccines are clustered together too. In this column, we can see many blocks. Then we try to use the embedding feature to classify vaccines as live or not. We divided the data into a training set and a test set, each comprising 50% of the total data. Then we build a neural network classification model. This figure shows the result the RC curves for train, test, and full dataset. The area and the curves are all about 0.8. We also use leave one out cross-validation to evaluate the performance of this model. It's very robust. We can see the sensitivity, specificity, and accuracy are all about 0.82. And the key findings include 3 aspects. First, LLM can efficiently extract adverse events with high accuracy. And the LLM method can identify the more differential AE profiles among vaccines. Also, we can find that LLM method can help to infer potential knowledge behind text, because we can classify the vaccines live or not. Future direction include apply the method to analyze other information on vaccines. And this step we have finished. We have developed a tool for ontology mapping. We can map the text to any ontology. And it can address the synonym issues. We just use the RAG theory to solve this problem. We can also apply additional machine learning method to analyze high-dimensional features. Also, we can use LLM to enhance the knowledge graph database and help find the potential biomedical information. Here is an example. Thank you for your time and attention, and welcome to visit our website. Thank you.
Okay, well, thank you very much, Dr. Wang, for great presentation. So I think we have 2 more minutes for Q&A. So please, does anyone have any question for Dr. Wang?
You know, I just wonder about, hi, this is Brian. Can you hear me okay?
Yeah, hi, Brian. Yeah, please go ahead.
Hey, Chuck, it's nice to see you.
Nice
and
to see
everybody. It was a nice, very important talk, you know, and I really appreciate it. You know, I just wonder about this privacy stuff. You know, I mean, it's just something that we all have to be concerned about when using the LLMs. I mean, one easy thing that I think we could do is that, you know, with the Llama 3, we've got it working on a laptop, you know, and it's not connected to the internet. So if there's any privacy concerns, that's one way we could deal with it. You know, it's just run it on a laptop with a GPU. Runs pretty good. Just a thought.
I totally agree. Dr. Wang, do you have any thoughts to share on that one as well?
Yes. I have tested the models smaller than 30 billion parameters can run smoothly on my my normal PC.
Right. Yeah. You know, you can, there's on Hugging Face, you know, you can optimize the size of the LLM for whatever PC you have. So it's really something that is quite good. And it's amazing how we do with fewer parameters, especially if we do the tuning. So it's really good. Thank you.
Thank you.
Yeah, thank you, Brian. Yeah, this one is, this one is like using the FDA package insert information. So yeah, of course, it's open source data, but the question you point like about the privacy definitely can be a concern. Yeah, it's a kind of new field. Exciting. Okay, I just stop here. So okay, great, Jiang, go ahead.